Good morning, everybody. Welcome back on your second day in our course. Just to be sure, I wrote down the name of the course that you're supposed to be in. If you don't recognize this course, you're welcome to stay, but you might be expecting something else. Right. So on the menu today is this lecture, Evolution and Homology. That Kim already started with a little bit yesterday. But before we dive into that, I have a few questions for you guys, because there's probably still people that need to find this building, need to find out how the elevators work, or if they work. We were in one where the doors wouldn't close and then the doors wouldn't open, and that was very interesting. And how to navigate this building is also something that you need to learn the first day that you're here. What I wanted to ask you guys was, how was yesterday? Great, thanks. Overwhelming, maybe? A bit? Frustrating? A bit? Not too much. Okay. Anything else that you want to share? Okay. So then, can I assume that it was good? Yeah? Well balanced. So a nice mix of great, overwhelm, and frustration. More or less as Kim commented you guys yesterday. She was talking about her lecture, but it goes for the whole course to some extent. I'll make the same disclaimer for my lecture of today. Many things will go very fast. Most of you guys have a biology background, so gene expression, evolution, and homology shouldn't be all new to you, but you might still like a refresher. If this is new to you, it will go very fast, right? So don't worry. There's plenty of time to fill in the blanks. You'll be doing the biology class on Friday. There's Wikipedia, there's Google, there's books, there's classmates that you can ask. Yeah? Okay, talking about classmates, yes. So, there's two things I need to do in addition to the lecture contents. One is to announce the group. Group assignments, who goes in which group. Lars is working on that. He will have it done before 11. He said probably around 10. So after the break, I hope to be able to tell you which group you're in and which group goes into which room. Which is convenient because that tells you where you need to go at 11. Right? If all else fails, if I forget everything or boss has a breakdown, which has never happened before, but you never know, just go to the room where you were yesterday. Yeah? So we have a backup plan in case whatever happens. Oh yes, the IT guys are working very hard to solve the very interesting problems that were cooked up for us over the holiday. You won't believe me if I tell you that we tested everything 2 weeks ago and it worked. Right? So one quote I got from one of the guys working at IT was, "We thought these systems were independent." Right? So ask anybody who has an IT background here, a science background here, whether that is, or animation over there, whether that is generally a valid assumption in any computer system. It's not true in biology, right? It's supposed to be true in computer science. We designed this stuff, and we should know better. Okay. Oh yes, and I should have done some time at the end, about 15 to 20 minutes, to introduce to you guys what you're going to be doing in the project. Right? So that's the menu. Right, then we can go. So, gene expression, evolution, and homology, and so it starts all with DNA, right? We have sequences, and basically the idea is that if we have the sequence information for an organism, for a species, we know everything there is to know about it. I'm not even sure if this is true in theory. Almost. It should be almost true. But it's definitely not true in practice. We don't really know. You can't look at a genome and say what it is. You compare two genomes. You can compare two genomes. And if you know something about one genome, you know, for example, it's E. coli, and the other one is similar, then what do you know about the other genome? It's probably also bacteria. Probably very similar to E. coli. It might be an E. coli strain or some next cousin of E. coli. I'm not a microbiologist, so I wouldn't know on the species tree what is closest to E. coli, but something close to E. coli. A lot of what we're doing in bioinformatics are just sophisticated versions of that. The phylogeny that Kim introduced yesterday is just a more sophisticated way of finding out who's closest to who, and therefore functionally and probably also morphologically most similar. So that's the best we can usually do. It's not just DNA, it's also proteins, right? So you know this is the central dogma of molecular biology. If you don't, now you do. DNA is transcribed into messenger RNA, which is translated into proteins. Proteins fall into a particular structure. They meet other proteins, other molecules. They either interact or they don't. This interaction either has an effect on the protein they interact with or it doesn't. And so life emerges. That's true that's actually true in practice, I guess. But it's much more complicated than I make it sound. Why is it so complicated? Because if it was this simple, we would have figured it out a century ago. What makes this so complicated?
Order regulation that goes.
Sorry?
Order of gene and protein regulation.
What is regulation?
There are different levels of like inhibiting or enhancing function or quantity in cell.
But it's inevitably molecules meeting, interacting, and either having an effect on each other or not, right? So you should all be able to describe this in terms of interacting molecules. You put them in your model, and then you have everything there. Where is the crux? Why this is difficult? Again, if this was as simple as I make it sound, it would have been solved a century ago.
It's highly dynamic.
It's dynamic, right? So two proteins don't just meet and then stay together forever. They divorce at some point. Could be sooner or later, right? And that might leave scars, or not. Yeah, but that's part of it. That's not all of it.
Cells are unbelievably chaotic, and we're just full up with stuff, and they will retain of those molecules and the ones involved in that.
Okay, so cells are chaotic, yeah, and lots of things are happening at the same time. Do you have any idea how many molecules are in a single cell? Let's take a small cell like an E. coli, right? A bacterial cell is pretty small, it's about a micrometer across. How many molecules? How many different types of molecules? How would you count the different types of molecules in an E. coli? We can start with the easy ones, the proteins. How do we know how many different kinds of proteins an E. coli can have? You measure the proteome. There's an easier way. You just look it up in a database. Where can we look it up? How do we know which proteins E. coli can make? Can we look at the DNA? Yeah. Yeah, that's proteomics. So that's when you really want to measure the cell, the proteins that are in the E. coli cell at this moment. But if I just want to have like a ballpark figure of how many different kinds of proteins E. coli could make. If I look in the genome and I count the number of genes, is that a good start? Yeah, so how many genes does E. coli have? 8,000, 12,000, something like that? Let's say 10,000. Yeah? Okay, so there's 10,000 different proteins. If I would do this for yeast, I count the number of genes in yeast. I think it's about 24,000-ish, something like that, if I remember correctly. It depends on the strain of yeast you're looking at, but let's say, let's take baking yeast. Does that tell us how many different kinds of proteins there are in yeast? Can be? No? What's different between yeast and E. coli? Splicing. So yeast is a eukaryote. It has a nucleus, but it has splicing mechanism. Splicing means the messenger RNA is edited in very interesting ways before it's translated into a protein, so you can make different kinds of proteins from the same messenger RNA. I know this is known stuff for most of you, but not for all of you. Okay, but let's go back to E. coli. So there's, let's say, 10,000 different kinds of protein it can make. What other kind of molecules are there in E. coli? Water? Yes, lots of it. Okay. By volume, actually, more water or protein? No. 70% protein. It's like a thick tomato soup inside. They're small, so you can't see it even with a microscope. You can't really see how thick it is. I might be over exaggerating a little bit. It's pretty full. Okay. Water, proteins. Ribosomes are made out of RNA and protein. Yeah, so there's RNA molecules in there. There's messenger RNA. There's transfer RNA. There's I don't know. Does E. coli have small RNAs? They probably do. Something like that. Right. Small RNAs, other stuff. Right. What other molecules?
Nutrients.
Nutrients. What? Yes. Okay. We're missing one important one. The DNA, right? That's also an important one. There's another important, very important one.
Lipids.
Lipids. Why are they important? Sorry?
Membrane structure.
They make up the membrane, right? So without lipids, you don't have a cell. Yeah. Well, without any of the other stuff, you also don't have a living cell. Okay. A lot of different kinds of molecules, right? There's different types of lipids. There's a huge host of metabolites, nutrients, and other stuff. There's proteins. Okay. So if we compare this to engineering, this is about as complex as a Boeing airplane. Yeah. So you can't see them from this room, but if you're in the other building, depending on which direction the wind blows, you'll see airplanes going in and out of Schiphol all the time. Yeah. So imagine each of those airplanes going in or out is about as complex as an E. coli cell. The advantage of engineers working with airplanes is that you can walk around in them and watch and look at every single component. And we have the blueprints and the maintenance manual and the instruction manual. Right? So we know how it works. Right? Okay, we don't have that for E. coli. You can't walk around in it, you can look at it, you can probe it, you can do knockouts, you can do all sorts of interesting experiments, you can do mass spectrometry to measure protein contents, you can do sequencing, we can't measure everything there is to know about an E. coli cell. And this is one of the simplest kinds of cells we have. Everything else is way more complicated. A typical eukaryotic cell, let's take a human cell, is about 1,000 times bigger and more complex than an E. coli cell. So in terms of engineering, it's about as complex as Schiphol. Is that the right direction? Yeah? It's about as complex as a whole airport, including all the airplanes flying in and out. So at that level of engineering, also engineers cannot predict how a system works. Think about IT. We thought they were independent systems. Yeah? You update something here, you update something there, and then lots of stuff in the middle breaks. And you have to figure out why. Because it wasn't independent. Yeah, it was supposed to be independent. Why is it not? Okay. So that's why it's so difficult to understand why cells are alive. Yes, it's this complexity. Good. I think we've covered a lot of the slides already. I tend to do this. So some students get upset, so that's why I mentioned it explicitly, that I tend to rush through my slides. That's because I were asking these questions and we've covered lots of stuff already. Right? So this one was already there, right? We've already covered networks, interactions, molecules, right? You somebody recognize? Who has seen this poster hanging on the wall of a lab? More than half of you. So it's known as the Boehringer Ingelheim Metabolic Pathways map. I used to have one, but I tossed it away last time I moved because it was hanging in the attic, getting dusty. Nowadays we have databases of this stuff, and we know a lot more than this. This is just touching the surface, but you can see there's quite a lot of stuff happening in there. Okay. Transcription regulation, I don't want to go too much into this, but transcription, almost everything in biology is regulated. Maybe everything, lots of stuff. What does regulation mean? It means if you have a gene and there's a transcription start site, and there's a polymerase binding site, the polymerase doesn't just go off transcribe this gene by itself. It needs to get a signal from a transcription factor, which might depend on other transcription factors binding, or it might depend on a particular nutrient binding to the transcription factor, which signals that we have to activate this galactose transporter because we're growing on galactose, and then we can get the galactose into the cell to grow. Right? If you think about it, and there are lots of these mechanisms. That's all called regulation. Okay. So and it's not simple. There's lots of elements involved in transcription. This is bacterial transcription. It's still relatively simple. Eukaryotic transcription has like 100 times more proteins in it. Okay. How does that look like to a bioinformatician? This is more the biophysics or the molecular biology view, right? This is molecules. You have a set of sequences. What do you look for when you want to understand how transcription like a regulation works? What can you see in the sequence?
Promoter sequences.
Okay, how do you recognize a promoter sequence?
Conserving it like data.
Okay, so you look for conservation. Why do you look for conservation?
Because they found out that it's very common that these sequences are in promoter regions.
That sounds a little bit circular. Right?
Yeah,
So
yeah.
you look for conservation because we know it's very common. But
Proteins can bind to those sites.
okay, so just take an example. So there's the title box over here, right? That signals that there's a transcription start site a little bit upstream. My molecular biology course is too long ago, so I don't remember exactly what the molecular function of the title box is, right? But it's something you can recognize. A little bit, downstream, there's a stretch. A little bit upstream, there's this MIG1 site, which is a binding site for a transcription factor that's called MIG1. I know that because it's annotated here. But the reason that it is annotated is that this one is a little bit better because you can see, I don't know if you can see it all the way to the back, but there's the block of C's here at the start, that's marked with stars, and the block is another set of C's and another set of T's over there. So this is the conserved part of this binding site. Why are these positions conserved?
Because mutation is constant, so if something is really vital for the cell, you want to conserve it, because otherwise you don't have a functioning gene.
So what happens if you don't conserve? Let's say this one is crucial for regulating, this is galactose, right? So what happens happens if you don't conserve, what could happen if you don't conserve this MIG-1 binding site over there?
You can't grow galactose anymore. And
Then what happens?
you either find a different substrate or perish.
Right, you die, right? You die. That's natural selection. It's a sort of a part of natural selection that people don't often talk about. If you get it wrong, you die. You're out of the game. Yeah? So why is there always a C over here? Is that because there was never a mutation at that position? Was there never a genome where there was an A, or a G, or a T over there? They've either died, or they were growing on glucose, or something else, right? Because you might not need this if you're growing on galactose. No, sorry, you might only need this if you're growing on galactose. Although usually, I mean, genes don't have a single function, so it's likely that this gene is also important in other conditions. It was named after galactose because we've seen that if you're growing these genes, these organisms, probably E. coli, on galactose, and you knock out this gene, they don't grow. Right? Oh, that is something to do with galactose. That's how we know this. Good. Now, think about regulation and transcription factors. What are transcription factors?
Proteins that act.
Sorry?
Proteins that activate polymerase.
Yeah, they're proteins, yes, and well, they have to do a couple of things. They have to bind into the DNA, and they have to make a complex that activates the polymerase. Yeah. But first and foremost, they are proteins. So that means there's a gene that encodes for the MIG-1 transcription factor. So now think about gene regulation. How can you see that you get a network of, or you can get a network of transcription factor genes regulating each other? Yes. Who has heard of this before? Transcription factor networks. Right? So we studied one a couple of years ago. I was going to say who recognizes it, but the hint is important. So this one comes from mice, and this is a set of transcription factors that are important for a differentiation of blood cells from the stem cell in the bone marrow to different types of mature blood cells like lymphocytes and B cells and lymphocytes, T and B lymphocytes, erythrocytes, and there's a couple of others. So, going from this, where you can see which molecule binds where to the gene of the other protein, you can simplify it by saying, oh no, I know SCL binds to the activator of ZFPM1, but to be able to activate ZFPM1, the GATA2 also needs to bind together SCL. Right? Actually, in most cases, SCL, or maybe all cases, let me check. In all cases, SCL can't activate a gene by itself. It always needs GATA2, or it needs GATA1. They're homologs, or parallels. Okay, so, and then you can study the behavior of this network. There's lots of different ways to do that. We did it with Petri nets, quite simple modeling formalism from theoretical computer science. You could say it's one step up from Boolean networks. So things can be on or off, but you can also say what are more on. You can assign discrete values to the interactions, and you can have positive and negative interactions, which is actually tricky to do in Boolean networks. It's much easier to do in Petri nets because two genes binding to the two proteins binding to the regulatory region of another gene might activate it, but two other proteins binding might actually repress the target gene. Right? So if you have GATA1 and ZFPM1 binding to the regulatory unit of GATA2, it's repressed. Okay. Now, a little bit more detail, but this is the stuff I already went through with you, right? So if you want to review this, it's in the slides. Okay, and there's lots of complications here. I don't want to go into. This is just a different way of writing a genome network. But here they're organized linearly as they are on the genome, but you can see that different genes are actually interacting. Where are we going? Yes. Yeah, this is for you guys to read if you need to review this. Now, so we talked a little bit about proteins, we talked about messenger RNA, we talked about gene transcription. What about DNA? What does it do? Proteins seem to do stuff. Transcription factors, they're enzymes, they make filaments, membrane receptors, lots of stuff. Messenger RNAs seem to do something. They carry information to the ribosome and other stuff. They are the ribosome. What does the DNA do? Sorry? It replicates itself. Almost. Sorry, there was another epigenetics. So DNA itself doesn't do much, right? To replicate, yeah. Yeah. So its main function is to store information. It's quite inert, actually. It's very stable. We can, well, not me, but there's people that can extract DNA from fossils. Not all fossils, but some. One of the tricks is to take hair. There's a ridiculously small amount of DNA in hair because it's basically just keratin. But there is some, and the advantage of it is that you can actually boil a hair, get rid of all the contamination except for the DNA that was inside it. But one way to get very pure DNA from mammoths, for example. Not fossils. That's when they're dug out of the ice somewhere in Siberia. There's also DNA that can be extracted from fossils. I don't know how to do it. So that's how stable DNA is. It's not. I mean, it's not undamaged over all these millions of years, obviously. Yeah, but it's very stable. But it has to. You have to be able to replicate it, right? And there's lots of stuff happening on and around DNA that sort of shape it, maybe also almost in a literal sense. And that has to do with what? There's actually two reasons for this. Well, reasons, I'm not sure that's the right word in evolution, but the two effects that you have from the stuff from epigenetics.
It's way too long to be in a single line in the cell, so you have to fold it so it can.
Yeah, it's packaging, right? And you get chromosomes and nicely packaged. Also makes it less sensitive to chemical influences and radiation and so on. So it preserves the information, helps preserve the information. Well, what happens if you fold up DNA?
Can't be processed. It can't be processed.
Yes, so you basically also at the same time we repress all the genes in the part of the genome that you've packaged. Yeah? You can't have it both ways. You can't read off a gene and have the DNA packaged at the same location. But there's lots of stuff happening there. So that was my next slide. And there's more stuff happening on. So there's acetylations and methylations. There's histone binding. These things are correlated. There's nuclear lamellar associations. There's other kinds of interactions between distant regions of the genome with each other that have to do with gene regulation, have to do sometimes with transcription activation, have to do sometimes with things we still have to find out. Okay, and yes, so this is yeah, one of the things that happens, for example, is you can have a whole series of transcription factor binding sites upstream of transcription start site, and but if they are bound to histones, they can't bind to transcription factor at the same time. Right? So they can be, and only the ones that are open, that are accessible will actually happen. So this is another layer, if you will, of regulation, yeah, amongst many others. I have hardly been on a conference in the last 20 years where I didn't hear about a completely new type of regulation in biological cells. Sometimes that was because I wasn't aware of that yet, but in most cases it was because nobody was aware of it yet, it was just discovered. And so there's things like, have you heard of transcription factories? Those are complexes of ribosomes and transcription factors and a couple of loops of DNA that apparently make the, or how to say this properly, that can enhance the transcription of a couple of genes sort of at the same time. Have you heard of ribosome regulation? There's lots of phosphorylations happening on the ribosomal proteins that seem to affect the binding affinity for different types of messenger RNA. So you can tweak a ribosome to prefer or dislike a particular type of messenger RNA. So after the messenger RNA is created, you can still tune how much protein you're going to get out of this, apparently. Right? This is just stuff I've heard of on conferences. And typically on a conference you bring the hottest, newest stuff, and I don't know what happened afterwards, if this was validated or reviewed or still open for discussion. But don't even if you've done 3 years of bachelor in molecular biology, don't assume you know everything about the cell. It's very unlikely that you already have to solve it. You might know everything that there is to know about the cell currently, but that's not the same as knowing everything about the cell. Okay. Translation. Yeah, this is for review. The codon table is for review. This is the codon table, but as a list format. And this is how ribosomes work. It's also for review. Good. I'm going very, very fast today. I'm going to see where I need to end up. I want a different view. So, hey guys, this is not the way you should give it up. That's there. Okay. So DNA is an information carrier, right? It's stable. It has an enormous information density. That's probably one of the main reasons why biological systems can be so complex. Is that within a single cell on the DNA, you can carry a lot of information. In 2010, when Jaap made this slide for the first time, it was estimated that worldwide, put it together, created about 3 zettabytes of data. And you need only 10 grams of DNA to store that, just like a tablespoon. In terms of DNA, that's still a lot. Right? If you would need to synthesize that amount of DNA, I'm not sure you could actually do that physically. I mean, DNA synthesis is still slow and expensive. But annual data production is growing exponentially. So for next year, you need almost 20 times as much. That's a teacup full of it. It's still a lot more condensed than if you would put it on SSD drives. I haven't calculated that. It would easily fill this room, possibly fill this building. So one of the really interesting things is that biological cells have this whole mechanism of writing and reading and copying DNA. So that's okay. And of course, I mean, we've all heard about next generation sequencing. The really cool thing about bioinformatics is that since about 20 years, we can very cheaply and efficiently read out DNA. Okay. I want to go somewhere from here. What was that? Oh yes, right. What are the 4 key elements of evolution? There's 4 things you need in the system to be able for it to evolve.
Selection.
Selection, that's Darwin. Yes?
Mutation.
Mutation, why?
Otherwise you won't develop.
You want to develop, okay?
To be able to select.
What happens if you start off with the population, you have selection, but no mutation? What happens to diversity? Sorry? You're thinking in the wrong direction. So you start off with a population of, I don't know, giraffes, right? They have different genomes. They evolve for a couple of 10,000 generations, right? But for some reason, there's no mutations happening. Very unlikely scenario. But we still have selection. So what happens to the diversity in the genomic diversity in this population of giraffes?
They'll probably have all the same DNA in the context.
Well, eventually, yeah. I mean, so depending on the conditions, some genomes will have a higher likelihood of survival. Others will have a lower likelihood, so they'll disappear. They'll die out. So the diversity can only shrink without mutation. So in order to invent anything new in evolution, you need mutations. Okay, so selection, mutation. There's 2 more to go. One was already mentioned earlier. Sorry? Replication. Yes, you need a copying mechanism. Because otherwise, how does the information go from one generation to the next? Number 4. So what's the mysterious one? What is the 4th one? Close. Sorry, what was it?
Radiation.
Radiation?
Variation.
Variation. If you have mutation, you'll get variation at some point. Or you have only a set of genomes that can't survive, then you're lost. You're on a dead end. No. Very important one.
Recombination?
No. It was already mentioned earlier, before in this lecture.
Conservation?
No, you get conservation because you have selection. It's so obvious, nobody sees it. No, you get complexity almost by default because that is apparently a way to survive better.
Exchange of genetic information.
But that's the copying.
Regulation? Regulation?
No, those are all effects of the.
It's
So,
just being able to produce substrate.
yeah, but that's the copying. So what are we copying? What are we mutating? And what are we selecting? Yeah, very close. You need a template. You need something to write your information in, or on, or with. Yeah? Something has to store the information to be copied. Something has to store the information to be mutated into different. Yeah? now let's go in a little bit back to the conservation. So, where does mutation happen? Sorry?
DNA.
It happens on the DNA, right? Where does the copying happen? It happens at the DNA, right? I mean, specifically, well, for sexually, for multicellular organisms, that's where you drink DNA for the offspring. I lost track. What mutations happen? Yes. Where does the copying happen? The template is the DNA, right? So where does selection happen? It happens in the environment. Close. Sorry? Sorry. On the phenotype. It's even simpler. Even simpler. So you're saying interactions of the organism, I'm just repeating, there's no interactions. So it's phenotype, interactions of the organism with the environment, it's all true. In the end, how does selection happen? I already mentioned before. Yeah, you survive or not. If you don't survive, it was not a good idea, your mutation. You thought of a brilliant, this C has to be an A. Nope, doesn't work. Not if you want to grow on galactose. Yeah? Or you do survive. Yeah? And of course, it's stochastic, there's a bit of luck involved, right? You might be able to get off a few generations on galactose without this particular transcription factor activating your galactose import mechanism. And then you find glucose again, and you're safe. Right? So it's not as straightforward as I might seem, but okay. So now we have these four elements. So phenotype is yeah, it's the expression of genes, but it depends. So it's if you dig down into these definitions, it's sometimes very hard to see what they actually mean. But for example, what does gene function mean? What is gene function? Take a slightly simpler one. What is protein function? Sorry? How it operates. Okay. So an actin protein. What is its function? It's simple, right? It makes a filament, actin filament. But it's not always that simple. What is the function of a transcription factor protein? Well, it has to bind to the transcription factor. Yeah, but it has to do a few other things as well. There's lots of proteins that are multi-domain, so they have a mix of different functions. Why are these brought together in one domain or one protein? Sometimes it's obvious, sometimes it's not. The bottom line of this is that whatever your phenotype or the functions of your protein, you can say conservation in sequences happens because function is conserved. Why is function conserved? Function is important to survive. This is a circular argument, right? So, protein function is whatever this protein does in the cell that allows the organism to survive. Whatever it is. It could just be in the way of something. It doesn't do anything, just there to prevent something else from happening. You never know what this something else is, because if you take it out, the cell is not viable, it dies. Yeah? So some proteins are more or less like that. So a protein or a gene function is whatever it's doing that helps the cell survive. And then it's a lot more complicated than that, because the cell needs to survive in many different conditions, many different environments. Okay. So we've covered this already. And oh yes, just one element there. In the end, it's all down to survival or not. So we see conservation in some places, but not all places of the genome. That is because there's a lot of things you can safely vary without jeopardizing your survival. Think of a car. You know the functions of the wheels. If you take off one wheel of a car, it doesn't drive. So that's why all cars have 4 wheels. They're not evolving systems, but the same logic applies. But you see cars in many different shapes and colors, because not all shapes, but many shapes will work. Yeah? And any color will work. Most people like dark blue or black colors, so most cars are like that. No, it's not because they drive better, because they sell better. That's a different function. There's lots of these things going on in engineering as well. Some of the reasons we build bridges the way we do is because we've always built them that way, and that's how bridges survive. Yeah? Because if you build a bridge in this way, it stays up. If you build it the other way, it breaks down. The next generation of bridges you're building are going to look like the one that stayed up. Nobody's going to build a bridge that looks like the one that just fell down. Okay, there's lots of these things happening all over. So, talking about this, it is an optimization process in a way. You've heard of the phrase the blind watchmaker, right? It's not a directed process. You try things, if it works, if you can, that's maybe not even the right phrase, if you can get away with it, then it's fine, right? So, in some cases, when you have like very strong competition between different microbes in a particular microenvironment, or different variations of the same species in one environment, you'll find that one particular variation dominates. Yeah? It doesn't mean that all the other ones are extinct. They might just be there a few percent of the population. They might turn out to be very important later on when the environment changes. It heats up or dries out or salt increases. Then there's this 1% that was not doing very well. Suddenly is much more favorable. Right? Ask the system biology people tomorrow. They do lots of research in microbial communities about this. Okay. So if you say the objective function that's optimized, well, yes, but it doesn't have to be optimal. It just has to be good enough to survive. There's nothing that tells evolution to be better than what you need to survive. A very simple example from ecology is how do you, as a gazelle, how do you avoid being eaten by lions? Do you have to be the fastest? No, it's good enough not to be the slowest. Yeah, the slowest gazelle gets eaten. The fastest one doesn't get eaten, but the slowest one also didn't get eaten. That's true for protein molecules. Okay, where are we going? We're going towards food break. Yes. Think about evolution over coffee.